This video demonstrates a procedure for isolating liver stem cells to study clonal expansion using CD133 cell surface expression following chronic liver injury. First, the liver is removed from a mouse with chronic liver injury and digested to produce a single-cell suspension. Red blood cells lysis buffer is then added to remove the red blood cells. And the solution is applied to a magnetic CD45 antibody-containing column to remove CD45 leukocytes. The CD45 depleted cells are then sorted to enrich CD133 positive liver stem cells. Finally, isolated CD133 positive liver stem cells are cultured and expanded in vitro. Gene and protein expression analysis demonstrate that the isolated CD133 positive stem cells are able to differentiate into both hepatocytes and cholangiocytes.
The main advantage of this technique over existing methods, such as co-localization of markers using immunohistochemistry techniques, is that single cells can be isolated for clonal expansion and functional analysis to confirm stem and progenitor cell phenotypes. Demonstrating the technique will be myself and Hein, a graduate student in my lab.
Prepare all buffers and media 24 hours in advance and store them at 4 degrees Celsius until use. All solutions, media, instruments, filters, and tubes should be sterile and